इस वीडियो में हम इनकम्प्लीट डोमिनेंस, कोडोमिनेंस, क्वांटिटेटिव करेक्टर और क्वालिटेटिव करेक्टर के बारे में पढ़ेंगे। तो चलिए शुरू करते हैं। जैसा कि हमको मालूम है कि मेंडल की वर्क जो 3 साइंसदान ने डिस्कवर किया था 1900 में, तो मुख्तलिफ किस्म के साइंसदानों ने मुख्तलिफ किस्म के जानदारों पर तजर्बात किए, जिसकी वजह से मुख्तलिफ किस्म के फिनामिना सामने आए, जिसमें से फहरिस्त है जीन इंटरेक्शन। So gene in various way interact with one another and produce the offspring is known as gene interaction. Or gene interact to modify the phenotypes is known as gene interaction. There are two types of gene interaction. One gene interaction in which one genes in their allele is responsible to produce the phenotypes. So we can say that it is the interaction within the genes. So it is known as intragenic interaction or allelic interaction. The interaction between the same allele which is present on the same locus is known as allelic interaction, and the other is known as two gene. Two or more than two genes interact with one another, and this type of interaction is also known as gene interaction. So when it is present between the two genes or more than two genes, it is known as intergenic interaction or non-allelic interaction. Interaction between allele of two different genes or non-alleles is known as intergenic two genes or non-allelic interactions. The example of one gene intragenic allelic interaction is incomplete dominance, codominance, multiple alleles, and pleiotropic genes, while the example of two genes are intergenic or non-allelic interaction is complementary gene, epistatic genes, supplementary genes, etc. etc. Then what is quantitative and qualitative character? As the name indicate, qualitative character, this character is not dependent on number of dominant allele, while quantitative character, quantity मिकदार को कहते हैं जैसा आपको मालूम है, और quality खासियत को कहते हैं, क्वालिटी को कहते हैं. So this character, quantitative character, is dependent on the number of dominant alleles. चलो हम एग्जांपल लेते हैं. अगर किसी भी मिसाल के तौर पर अगर हम पी प्लांट्स में कैपिटल टी वाले इस जीनोटाइप को ले लेते हैं, उसमें डोमिनेंट एलील 2 है और इसकी हाइट 10 सेंटीमीटर होगी. इट हेटेरोजाइगस कंडीशन में डोमिनेंट जीन्स 1 है और इसकी हाइट भी 10 सेंटीमीटर होगी. और यहाँ पर डोमिनेंट जीन एब्सेंट है तो डोमिनेंट अलील भी एब्सेंट है, तो हाइट 5 सेंटीमीटर है. और इस तरह क्वांटिटेटिव करेक्टर में यहाँ पर 2 जीन्स है तो यहाँ पर 10 सेंटीमीटर जो है ना हाइट है. यहाँ पर एक डोमिनेंट जीन है तो 7.5 सेंटीमीटर जो है ना वो हाइट है. और होमोजाइगस रिसेसिव में डोमिनेंट जीन एब्सेंट है तो वहाँ पर 5 सेंटीमीटर है. So the character which is dependent on the number of dominant allele is known as quantitative character, and those character which is dependent on number of dominant alleles is known as qualitative characters. Now we'll come to explain what is incomplete dominance. Incomplete dominance is a phenomena which is not discovered by the Mendel. It is discovered by Karl Korens of Germany, and it is known as post-Mendelism because it is discovered after the work of Mendel. It is that's why it is known as post-Mendelism. So, incomplete dominance, also called monogenic quantitative inheritance. It is also known as phenotypic blending, exception of Mendel law of dominance. इसको exception of Mendel law of dominance भी कहते हैं. In this interaction, dominant allele is not fully dominant to a recessive allele. As the name indicates, incomplete dominance is those form of dominance in which the dominant gene is not completely dominant over the other genes or recessive genes. So this type of dominance is known as incomplete dominance. So in heterozygous condition, an intermediate phenotype appear. Thus, if one hybrid does not resemble to any of the parents, in this inheritance, phenotypic and genotypic ratio are obtained. In incomplete dominance, the phenotypic ratio will be equal to genotypic ratio. As we know that it is discovered by Karl Korens of Germany in flower of Mirabilis jalapa, which is also known as poroclac plants or gulebans or guliabbasi. So flower color in Mirabilis or poroclac plant is red, while white color flower is also present in Mirabilis jalapa. When we cross red color flower with white color flower, it produces the pink color flower in F1 generation. Upon selfing for the production of F2 generation, we cross F1 with F1. So you clearly see here that one red, two pink, and one white is produced. So the phenotypic ratio is 1 ratio 2 ratio 1, while genotypic ratio is also 1 ratio 2 ratio 1. So phenotypic ratio will be equal to genotypic ratio. Remember, in complete dominance, the genotypic and phenotypic ratio is not the same, while in incomplete dominance, the genotypic and phenotypic ratio is the same. We take another example. So flower color in Antirrhinum majus are, which is also known as snapdragon, are dark flower. Red color flower is crossed with white color flower. So the flower which is the offspring which is produced in one generation have all pink color. Upon selfing, it produces one red, two pink, and one white color flower. So the phenotypic ratio and genotypic ratio is also the same. Feather color and undulation fowl. ये खास किस्म के परिंदे हैं. इसमें ब्लैक और व्हाइट कलर के परिंदों को अगर हम एक दूसरे के साथ क्रॉस करते हैं तो जो F1 जनरेशन में जो औलाद उसकी पैदा होती है तो वो सब के सब जो है ना ऑल ब्लू कलर की होती है. और F2 जनरेशन में हम ब्लू और ब्लू एक दूसरे के साथ क्रॉस करते हैं तो इसमें एक ब्लैक, दो ब्लू और एक व्हाइट आएगा, जो कि जिसमें 1:2:1 phenotypic और genotypic रेशियो भी सेम होगा. साइज ऑफ स्टार्च ग्रेन इन सीड ऑफ गार्डन पी प्लांट्स. लार्ज साइज स्टार्च ग्रेन और स्मॉल साइज स्टार्च ग्रेन, अगर हम इसको क्रॉस करें तो मीडियम साइज स्टार्च ग्रेन जो है ना इसकी ऑप्शनिंग में आएगा. सो ये भी हमारे साथ इनकम्प्लीट डोमिनेंस की एक बेस्ट एग्जांपल है. So with respect to size of starch grain in pea plant, large size starch grain plant is crossed with medium size starch grain and produce 100 offspring. Find out number of large, medium and small size respectively. So as we know that large size starch grain is represented by capital B and small b, and medium size is represented by capital B and small b. So upon crossing, large should be 50 and medium should be 50 and small should be absent. And mirabilis and red flowers produce F1 generation. Now F1 generation is test crossed and produce 200 offspring. How many of them are red, pink and white flower? As we know that red is crossed with white, so it produces the pink color. Upon test cross with recessive parents, it produce one pink and one white. So here the in test cross, the ratio of phenotypic and genotypic is also the same. Codominance, exception of Mendel law of dominance. As the name indicate, जिस तरह के हम माशरे में मुख्तलिफ किस्म के अल्फाज इस्तेमाल करते हैं, कोऑपरेशन, कोऑर्डिनेशन, को एजुकेशन, तो यहाँ पर हमारे साथ जेनेटिक्स में कोडोमिनेंस है. सो एक्सेप्शन ऑफ इसको also known as exception of Mendel law of dominance. In this interaction, in heterozygous condition, both alleles are equally dominant and show their independent expression. So in heterozygous condition, an intermediate phenotypes are not appear, but both the genes express himself and their offspring are F1 generation. Then such type of dominance is known as codominance. Again, I am defined codominance is a type of interaction in which both the alleles express himself in F1 generations. So it is known as codominance. Thus, F1 hybrid resemble to both of the parents, and this inheritance phenotypic and genotypic ratio are obtained here same. So coat color in cattle, black is crossed with white. So in F1 generation, roon color is obtained. Roon mean इसके जिस्म पर मुख्तलिफ किस्म के दाग और धब्बे होंगे. Upon selfing, when we cross sisters and brothers within the cattle with one another, so it produces black, one black, two roon and one white. So here the phenotypic as well as the genotypic ratio is the same, and this is the example of in codominance. AB blood group is also in human is the example of codominance. A person having blood group A is married a girl having blood group B. So all of their offsprings should have AB blood group. Either their daughter and son will be blood group of AB. So it is also a good example of codominance. But remember here the blood group A individual as well as the blood group B individual will be homozygous, so mean the same. Sickle cell anemia is also a best example of codominance. Sickle cell anemia, it is also known as drepanocytosis, in which lack of red blood cells, lack of hemoglobin, and lack of oxygen is present. Point to keep. Codominance is here both the alleles express himself in F1 generation. Here the phenotypic and genotypic ratio will be the same. In complete dominance, the phenotypic and genotypic ratio will not be the same. While incomplete dominance, here the phenotypic and genotypic ratio will be the same. So please remember it.